Hello everyone, welcome to this tutorial. Today we will do uh, DNA-seq analysis with Python and Google Colab. This analysis totally done in the Google server. So don't need to install any software in your in-house laptop to do this analysis. And this all the all the Google Colab notebook that we are using today has been given into the description. You can use it from them directly. So for this analysis, first we have to convert the uh, feature count output into a CSV file, and for this we will use this code. And after that, when we convert all those into a CSV format file, and after that we will combine them with this code into a single CSV file. So first we have to upload all the data. Okay. This is the feature count output. We upload it here. Okay, all the file have been uploaded. Now we need to copy the path of the first folder according to your data set. Which one of is your first data? Copy it here. So copy it and paste it and change the name of the output file. This is very important. Do not forget to do this. Okay. You'll see a CSV file will appear here. Okay, here. Okay, like this we can make for the other also, because they have the same naming pattern. I can do this. Okay, we have done converting all the feature count output into a CSV file. Now we need to run this code. This code will detect all the CSV file in this sample data folder and combine them into a single file according to their serial, or according to the serial. Okay, file has been generated. This is the file. We have to download the file. Okay, file is downloaded. So you can see according to their uh, serial, they have been sorted here. Like this 35, this 36, this is 37. Okay, you can see it here. This is 38, this is 39, and final one is 40. Okay, but we have to trim it. This portion we don't need. Trim this and this, and later on do this. 40, 39, 38, 37, 36. Save it and close it. Now go to second notebook. This is for differential expression visualization with PyDSec 2. Okay, now you can simply download folder. Okay, uh, this one we downloaded. We load this data here. Okay, first we need to install the PyDSec 2. It will take some time. Later we have to load this. Okay, now we have to load our data. Copy path and paste it here. Now run this code. It will load our data. Okay. Set the index to the gene ID. Now we filter all the zero count from the sample. Okay. Mm. This will reverse the column and row. Now we load the metadata. Okay, we loaded the metadata to our count data. See, according this metadata already set it according to my dataset. You need to change it whichever dataset you are using according to the control and treatment and whatever condition you are having. Okay. Metadata loaded, and now we set the metadata to our sample. Now we need to make a DSEQ dataset. Our dataset has been prepared. Now we will apply DSEQ to analysis into this dataset. It will take some time. Okay, our differential expression analysis has been done. Now we can check our differential expression dataset. Just simply read DTS. It will show the dataset to us, and you can use this different type of command to further investigate your dataset. Now, okay, state summary. Okay, this is also done. We can now we can save our now in this code we'll save the DSEQ result into the RAS dataset. Okay, now with this code, what will this code do? This code will save the DSEQ data frame into RAS into DSEQ to result treatment versus control CSV folder that been yes, this is here generated. You can download it for further reference and keep it here, and it's here. We can continue our analysis. Now we need to annotate the data set. So we will use biomat to do that. So let's install biomat. Okay, this is done. Now we load biomat along with required feature. Biomat and required tools has been loaded to the Google Colab. Uh, sorry, that's fine. Now annotate the dataset. Okay, annotation is all. Now annotate the dataset to DSEQ to gene list. It will take some time. Okay, this is also done. Annotation is also done. Now we combine these two dataset. Okay, we can see that uh, against this gene ID, what is the gene symbol and gene description has been added to the dataset. Now we save this dataset as CSV. Okay, okay. This data is this data is also generated. We need to save download this dataset because we need to use this in gene set enrichment analysis and KEGG pathway analysis. Okay, there is no extra symbol and description column here, so we don't need to use this code. Okay. Now we have to filter a significantly differentiate express gene. Just run this and it will be done. Now, significant data frame has been generated. Now we can run this and check if this. Okay, this is fine. Okay, you can also check it from here. Look here, 27,688 rows and 8 columns. And here when we filter for the significant data set, now rows has been reduced into 9,433 rows and 8 columns. That will be constant as usual. Okay. Okay, now we'll do principal component analysis. So do this with scanp. Now install scanp. It will take a little bit of time. Sometimes it requires. Okay, if we again need to do some. Okay, if we need to reset the dataset, we need to do some analysis again, because when we reset a session in Google Colab, it actually remove all the previous data which has been saved in the Google in that particular session. So I'll do it again. For this time, I'm just pausing the video and I will start from the same place again. So what now, when once this been done, we do not need to again reinstall the scanp or whatever feature we are using. So this is fine. Now we need to import the scanp. Okay, then perform principal component analysis into the differential dataset and plot. Okay, now it's been plotted, so you can download it, save as, uh, give it a name. God, okay. Grapher, this is from Scanpy. We are using to make Heatmap. Okay. So Grapher is a dataset that we use for the Heatmap cluster heatmap. So just run this code. After all this, it will create the heatmap. Okay, this is the global heatmap. So you can see a very good, very beautiful heatmap here that we can easily change in which gene is differentially expressed in which part. So okay, this is check the data set. Okay, create a log p expression data set from the for the target gene. What will this do? If you have if you have some target gene that you need to check in the test sample if they're differentially expressed or not, compared to the control, you can use this. So this gene list, you can denote your gene here, which gene you need to check. Okay, so it will create the data set that contain the data from all those for all these genes in those data sets, RNA-seq data set. Now we need to annotation data frame. So this has been created. This is called this. Okay. Now we need to replace this in the count data and data frame. Replaced. Okay, gene ID has been replaced with their symbol. Okay, after that we can visualize it. Please. Okay, you can download it and you save images. Okay. Now by using sandbombings we can plot very beautiful volcano plot, and which is very easy. We just need to use the RS dataset. This is Differential Express dataset to do this. And this one. Okay, by the way, this RS dataset is annotated. The annotated dataset we need to use. This is this symbol column. Okay, we need to import Volcano from the sandbombings plots. Check the RS dataset. Okay, this is, and just simply run this code. Which will produce this uh, volcano plot. So I can save it. Okay, now we are done with this notebook. Now we can move to our gene set enrichment analysis. Okay, let's sorry for that. Okay, so our notebook is getting connected from with the server. We can close the previous one. Here we have to upload annotated dataset. This one. Let's get uploaded. Let's load the copy the path and paste it. Load the data frame. See the data frame, it is fine or not. Okay, it is fine. From here we are actually dropping this description column that we don't need for further analysis. Data from the, we check that. Okay, the description column is gone from the data frame. Now we need to rank the gene. Both up regulated and down regulated gene will be ranked according to their expression profile. From there we will just keep gene and their rank. Next we need to filter out this INF, this. So just cleaning the dataset. Now we need to order the gene. Now this gene set enrichment analysis will be done by G profiler. Install the G profiler. Okay, and then call the G profiler. Now run the G profiler against our data frame, and this will create this uh, gene set enrichment analysis result data frame. Okay, it will take a little bit of time. Okay, gene set enrichment analysis has been done, and the uh, result data frame has been created. Now, this in this code, we are actually calling first 20 data of this GSEA result data set to show. Okay, this is fine. Now into we have to save it. What will this do? This will create a CSV file of this particular data set. It will show we can, okay, this will create it. Okay, now we'll use Plotine. Actually, Plotine is a ggplot implementation, Python implementation of ggplot. Uh, okay, this code will create a beautiful bubble plot from this particular data set. We can see response to stimulus. Okay, here if we see the color, this actually denote the significance of that particular data frame. And if we see the bubble size, this how many the number of genes actually associated with that particular particular data frame, particular responses. So okay, and you can save it. Now the bar plot. For bar plot, simply run this code. It will create the bar plot, and again the color is actually denote the significance, and the length of the bar uh, represent the number of the genes or counts associated with that particular response. Okay. Okay, for enrichment map, use this below pipeline. Similarity. Uh, Network is for making the network between all these all these gene set, or all these gene set that has been responsible for this various type of responses. Okay, now we need to plot it. This is the plot. You can simply save it. Now for the KEGG pathway analysis, we will use this one. Connect the connecting. Okay, it's connected. Sorry. Okay, okay. Upload the annotated data set. Copy path and paste it here. Sorry. So again we will do this KEGG pathway analysis with the G profiler. Load the G profiler to the Google Colab. In the next code, we are doing KEGG pathway analysis against uh, Arabidopsis thalaina against our dataset that we loaded. Now run the dataset, and this code will produce this KEGG res data frame, which has been shown here. Okay, now it visualize this dataset. Run this to visualize, basically to form the bubble plot. So this is the bubble plot of the KEGG pathway analysis. So you can see that API transporter, especially this metabolic pathway. This is a very vague name, but basically this uh, okay, okay. Uh, can save it. KEGG. Okay. So link for all this Google Colab notebook is given into the description. You can use it from there, and that's it for today. Thank you for watching this video. Bye bye.